close your eyes if you like. Let your shoulders fall gently away from your ears, and let your hands rest open and still against whatever warmth surrounds you. There is nothing you need to do right now. There is nowhere you need to be. The day has already passed through you, and all of its noise is beginning to soften at the edges, growing quieter and further away with each slow breath you take. We are going to travel together, you and I, back through time, not quickly, not urgently, but the way a river moves through a wide, flat plain, with patience and with purpose, and with no particular hurry at all. We are going to drift back to a part of the world you may never have visited, to a season that has long since passed, to a place called Yambuku in a country then known as Zaire, in the late green summer of 1976. The trees there were very tall, the air was warm and heavy with the presence of the forest, and the mission hospital at the center of the village was a quiet place, white-walled and modest, staffed by Belgian nuns who had given their lives to caring for people in one of the most remote corners of the earth. If this voice is helping you rest, consider subscribing to Plague Lullaby, but only if you are still awake to do so. For now, simply breathe and let the story carry you the way the current carries a leaf, gently, without resistance, downstream. It is worth beginning, as all such stories begin, not with a catastrophe, but with a single unremarkable moment. The kind of moment that, in hindsight, historians would later examine with great care, turning it over like an object found in the earth, wondering how something so small could contain so much consequence. The year was 1976, and the month was August, and in the village of Yandongi, not far from the mission at Yambuku, a man whose name the earliest records preserve only partially went about the ordinary business of his life in the equatorial forest of northern Zaire. He had been traveling, some accounts suggest, moving northward through the region, eating the varied foods available to travelers in that part of the Congo basin, including, as was common and unremarkable at the time, the meat of wild animals that inhabited the dense surrounding forest. He was not a remarkable man, so far as the historical record is concerned. He was not famous. He was not powerful. He was simply a person living in the world, making his way through a landscape of extraordinary green richness, under a sky that must have seemed on certain mornings to contain the whole of possibility within its wide blue reach. And yet what happened inside his body during those late summer days in 1976 would, within weeks, send an invisible signal rippling outward from that remote forest in ways that the world had not yet learned to read, and that the world was not yet, in that particular summer, prepared to understand. The man arrived at the Yambuku Mission Hospital in late August 1976, presenting symptoms that the Belgian's nurse and the attending physicians recognized immediately as consistent with malaria, which was then, as it is now, extraordinarily prevalent in that region of equatorial Africa. He was feverish and fatigued, and the clinic, following its established routine, administered to him an injection of chloroquine, which was the standard treatment for malaria at the time, delivered using one of the 5 glass syringes that the mission hospital kept in rotation for its outpatient clinic. Those syringes, as was then standard practice in many under-resourced rural clinics, were rinsed in sterile water between patients, but were not individually sterilized before each use, since sterilization took time and resources that the mission did not always have in abundance. The man recovered from his immediate symptoms and returned to his village, and for a brief period, it seemed as though nothing extraordinary had occurred. But he had introduced into the hospital, through whatever transmission had reached him in the forest, a virus that no laboratory in the world had yet formally named, described, or catalogued. Over the days that followed, other patients who had received injections at the same outpatient clinic began to fall ill with symptoms that progressed far beyond what malaria typically produces. And the nurses who had cared for these patients, women who had traveled from Belgium to serve in this remote mission, began, one by one, to feel the first warmth of the fever working quietly within them. Within the first week of September 1976, it became clear to the staff of Yambuku Mission Hospital that something unusual was unfolding in the wards and in the surrounding villages, with a pace and a severity that went beyond anything in their previous experience. Patients experienced high fever, and after several days, a number of them developed conditions affecting multiple organ systems, with fluid accumulating in ways that the standard treatments of the time could not address. The nurses who had continued to provide care fell ill themselves, and Sister Miriam Zecker, Sister Beata de Fays, and Sister Augustina Deknoit, among others from the small Belgian staff, became patients in the same hospital where they had for years been caregivers. The hospital, which had been a source of healing and trust for the surrounding communities, was now understood with growing and terrible clarity to be the point at which the illness had spread most efficiently, carried on the glass and metal of the syringes that moved from arm to arm without adequate cleaning between patients. Local Zairean health authorities were notified, and word traveled, as it always does in circumstances of sudden medical alarm, through the slow but deliberate channels of regional government and international health organizations. By the time the Zairean Ministry of Health in Kinshasa received word of the outbreak, the number of cases in the Bumba zone of the Equateur region had grown substantially, and a kind of quiet, heavy understanding was beginning to settle over those who were hearing the reports that this was not malaria, and it was not typhoid, and it was not any of the familiar fevers that had circulated through Central Africa for generations. During this period, a blood sample was collected from Sister Miriam Zecker, one of the Belgian nuns who had fallen ill, and sent in a thermos packed with dry ice on a flight to the Institute of Tropical Medicine in Antwerp, Belgium, which was the natural recipient of specimens from the Congo, given the long historical connection between Belgium and its former colony. The sample arrived at the Institute in early October 1976, and it was there that a young Belgian scientist named Peter Piot first encountered the blue thermos and the carefully packaged vials within. Piot was 27 years old, newly trained, and working at the Institute in Antwerp, where his supervisor, Dr. Stefan Patin, had received the samples for analysis. When Piot examined the specimens under an electron microscope, he found inside them a virus of extraordinary and unfamiliar shape, long and filamentous, twisted in a way that resembled the Marburg virus, which had itself caused a small and devastating outbreak in Germany and Yugoslavia in 1967 among laboratory workers who had been exposed to tissues from African green monkeys. The virus Piot was looking at shared the characteristic thread-like morphology of Marburg, but showed no reaction to the antibodies that would have identified it as such, meaning this was not Marburg, but something related to it, something new, something that the world had not formally seen and described before. He and his colleagues sent word to the Centers for Disease Control in Atlanta and to the World Health Organization in Geneva, and the movement of scientists that followed, carefully, deliberately, over the course of days and weeks, would eventually bring a small international team to Yambuku itself to begin the careful, patient work of understanding what had happened and what it might mean. The Centers for Disease Control in Atlanta, which in 1976 was already the preeminent institution in the world for the identification and response to novel infectious diseases, received word of the Yambuku outbreak through its established networks of global disease surveillance and dispatched a small team of scientists that included Dr. Carl Johnson, a virologist of considerable experience, who had previously identified the Machupa virus in Bolivia, and who would later receive credit for naming the new virus found in northern Zaire. Johnson was a careful and methodical scientist, shaped by years of fieldwork in remote and difficult environments, and he understood from the earliest reports what many others did not yet understand: that the pattern of transmission, the severity of the illness, and the geographic clustering of cases in and around the mission hospital suggested a pathogen of significant power that was nonetheless spreading, at that stage, primarily through direct contact rather than through the air or through casual encounter. The WHO also convened an international commission that brought together scientists from Belgium, France, the United States, and other nations, and when this commission arrived in Yambuku in October 1976, they found a community profoundly altered by weeks of illness and fear. The hospital shuttered, the roads into the affected area, under a kind of informal quarantine that had been established by a combination of local initiative and government directive. The commission set up a simple field laboratory in the mission, and the work of interviews and specimen collection and epidemiological reconstruction began. Slowly, carefully, systematically, in the way that good science always proceeds, without hurry, without assumption, following the evidence wherever it quietly leads. The International Commission's field investigation in the Bumba Zone in October 1976 produced, over the following weeks, a picture of the outbreak that remains one of the most carefully documented early responses to a novel pathogen in the history of infectious disease. The scientists fanned out across the affected villages, recording cases, tracing contacts, mapping the movements of the ill, and building a detailed picture of who had fallen sick, and when and through what apparent connection to others who had also fallen sick. What emerged from this painstaking work was an epidemiological portrait of extraordinary clarity. The great majority of cases were connected either to the Yambuku Mission Hospital itself, where the inadequately sterilized syringes had served as the primary vector of transmission from patient to patient, or to family members who had cared closely for the ill, particularly in the intimate context of washing and preparing the bodies of those who had died in the traditional Congolese manner of burial preparation. The virus, the commission determined, required direct contact with the bodily fluids of a person who was already ill in order to pass from one person to another, and this finding, quiet and precise and fundamentally important, meant that the outbreak, devastating as it had been within the hospital and within the families of the affected, was not capable of spreading indefinitely through casual community contact in the way that influenza or measles can. This finding would prove crucial not only to the control of the 1976 outbreak, but to the conceptual framework through which scientists and public health workers would understand and respond to every Ebola outbreak that followed over the next 4 decades. By late October 1976, the outbreak at Yambuku was approaching its natural conclusion, not because of any treatment that had been administered, for there was no treatment and there would not be one for many years, but because the hospital itself had been closed. The syringes had been taken out of circulation. and the quarantine measures that had been put in place around the Bumba zone had interrupted the chain of transmission with quiet effectiveness. The final count of the outbreak, as recorded in the formal report published in 1978 in the Bulletin of the World Health Organization, was 318 cases and 280 deaths, a case fatality rate of approximately 88%, which was and remains one of the highest ever recorded for a naturally occurring outbreak of any disease, and which spoke to the extraordinary severity of the illness in the absence of any supportive care beyond what a small, understaffed, and overwhelmed mission hospital could provide. The Belgian nuns who had survived wrote letters home, and the doctors who had worked in the field wrote their reports, and the specimens that had been collected were taken back to laboratories in Antwerp and Atlanta, where the slow, careful work of characterizing the new virus would continue for months afterward. It is worth noting, in the way that one notes something that feels both small and enormous simultaneously, that 280 people had died in a remote corner of the world, that most of the world would never visit, that their deaths had been recorded with care by scientists who understood the weight of what they were documenting, and that those deaths had given the world its first formal encounter with a virus that would remain present in that part of the world, quiet, elusive, occasionally devastating, for decades to come. The naming of the virus, which occurred in late 1976 and early 1977, as the scientific community worked to formally describe the new pathogen, was itself a moment of quiet historical significance, though it was accomplished not with ceremony but with the pragmatic urgency of scientists who needed a name and needed it quickly for the purposes of publication and communication. Carl Johnson, the American virologist who led the CDC team, has been widely credited with the decision to name the virus after a river, the Ebola River, that flowed through the general region of northern Zaire where the outbreak had occurred. The Ebola River was not the closest river to Yambuku, as Peter Piot would later acknowledge in interviews and in his memoir published decades afterward. The team was exhausted, working long hours in a foreign country under difficult conditions, and the Ebola was simply the river that appeared on the maps they were consulting and seemed sufficiently nearby to serve the purpose of geographic identification. Pio later reflected, with characteristic gentleness, that the naming was perhaps not perfectly precise in geographical terms, but that names, once given to viruses, tend to persist regardless of their exact accuracy, and so the Ebola River, dark and quiet and far from famous, gave its name to one of the most studied viruses in the history of medicine. The name was formally adopted in the scientific literature in 1977, and the virus was classified within the family Filoviridae alongside the Marburg virus, the two of them forming a small, distinctive, and deeply studied family of pathogens united by their shared morphology and by the particular and devastating manner in which they interact with human physiology. Meanwhile, and it is important not to pass over this detail too quickly, a separate outbreak had been occurring at almost exactly the same time in southern Sudan, in a town called Zaire in what is now South Sudan, where workers at a cotton factory began falling ill in June 1976, actually predating the Yambuku outbreak by several weeks, though the two were discovered and investigated more or less simultaneously by the international health community. The Sudan outbreak produced 284 cases and 151 deaths, for a case fatality rate of approximately 53%. Severe, deeply sorrowful, but somewhat lower than the Zaire strain. a difference that scientists would later attribute to subtle variations in the viral genome between the two strains, which were classified as separate species, Ebola virus Zaire and Ebola virus Sudan. The Sudan outbreak was investigated by a separate WHO commission, which found a similar pattern of hospital-based transmission amplified by inadequate infection control alongside transmission within households among family members who had cared for the ill. The two outbreaks, occurring in the same season on the same continent, caused by related but distinct viruses that had apparently emerged independently into human populations, served as a kind of double introduction. A first chapter written in 2 columns simultaneously that gave the world its initial, hard-earned understanding that this was not a singular event but a category of event, not one virus but a family, not one outbreak but a pattern that would recur across the decades with quiet and unpredictable regularity. The equatorial forest in 1976 had revealed something that it had been keeping in all probability for a very long time. And in the years that followed, scientists would spend considerable effort trying to understand exactly where, in the deep biological complexity of that forest, the virus spent its time between the moments when it entered the human world. The years immediately following the 1976 outbreaks were, in terms of Ebola specifically, a period of relative quiet, not because the virus had disappeared, but because the conditions for a significant human outbreak had not yet reassembled themselves in any of the ways that the 1976 conditions had, and because the virus was behaving, so far as anyone could tell, in the manner that viruses like it tend to behave in the intervals between human outbreaks circulating at low levels in whatever animal population served as its long-term natural reservoir, causing disease perhaps individual animals without sufficient concentration to reach the threshold at which human infection becomes likely. Scientists in the years and decades following 1976 spent considerable effort trying to identify the natural reservoir of the Ebola virus, which is to say the species in which the virus lives, without causing significant illness to the host, the species that maintains the virus between human outbreaks, the way a library maintains a book between readings. Early investigations in the late 1970s and 1980s tested hundreds of animal species in the affected regions of Central Africa. Bats, rodents, primates, insects, birds. But the results were inconclusive, and the reservoir remained formally unidentified for decades, a mystery that sat at the heart of Ebola's ecology, and that made prediction and prevention of future outbreaks significantly more difficult than they might otherwise have been. The forest was generous in many respects, but guarded in this particular one, and the question of what species carried the virus through the long, quiet years between outbreaks would not receive a satisfying, if still provisional, answer until the early years of the 21st century, when evidence began accumulating that fruit bats of the family Pteropodidae were the most likely candidates. In 1979, a second outbreak of Ebola Sudan occurred in Zaire, in the same location as the 1976 Sudan outbreak, affecting 34 people and killing 22 of them, and confirming for epidemiologists what the pattern of the 1976 events had suggested, that this was a virus capable of reappearing in the same communities. Years after its first visitation, perhaps because the reservoir was geographically stable and the conditions for spillover into human populations were determined by factors that science had not yet fully articulated. The 1979 outbreak was smaller than its predecessor, was contained through the same combination of isolation quarantine, and infection control that had worked in 1976, and left behind it the same questions that 1976 had left behind. Where does the virus go when the outbreak ends? And why does it return when it does? And what determines who is exposed and who is not in the complex ecology of a forest, where humans and animals have shared the same space for thousands of years? These were patient questions, the kind of questions that good science approaches slowly and carefully over years and decades, accumulating evidence through fieldwork and laboratory analysis and epidemiological investigation, never rushing to a conclusion before the evidence is sufficient to support one. And it is worth pausing here, in the quiet of this narrative, to appreciate the particular dedication of the scientists and public health workers who spent years in the field, in difficult conditions, asking these questions on behalf of all of humanity, often without recognition and frequently without adequate resources. The long interval between the 1979 and Zaire outbreak and the next significant emergence of Ebola in the human world lasted for more than a decade, and during those years, the scientific understanding of the virus deepened in the controlled environments of high-security laboratories in Atlanta, Antwerp, Marburg, and elsewhere, where virologists worked with Ebola under the strictest biological containment conditions available. Conditions designated Biosafety Level 4, the highest level reserved for pathogens for which no vaccine or treatment exists, and for which exposure carries a significant risk of severe illness or death. Research published in journals through the 1980s began to clarify the basic biology of the Ebola virus, its structure as a single-stranded RNA virus, its replication mechanism within host cells, the particular way in which it interacts with the human immune system to produce the cascade of effects that characterize severe Ebola virus disease, including high fever, profound weakness, and in some cases the internal and external bleeding that had given the illness its original, and now somewhat outdated designation as a hemorrhagic fever. Scientists discovered that the virus is exceptionally good at evading the initial immune response, entering cells through a mechanism that involves a protein on the viral surface binding to receptors on the surface of certain host cells, particularly macrophages and dendritic cells, which are among the first responders of the immune system, and which the virus, in effect, subverts and repurposes for its own replication, using the machinery of the immune response itself as a vehicle for its own spread through the body. The social and cultural weight of the 1976 outbreaks settled over the affected communities in ways that extended far beyond the immediate medical consequences of the illness itself. And this dimension of the Ebola story, the human dimension, the dimension of meaning and memory and community response, is worth moving through with the same care and attention that we give to the scientific and epidemiological dimensions, because it is in this dimension that the full complexity of what it means to live through such an event becomes most visible. The villages of the Bumba Zone had experienced, within a period of less than 3 months, the deaths of a substantial fraction of their members, not from war or from famine, but from an illness that arrived without warning, progressed rapidly, and for which no cure existed. The hospital that the community had trusted as a source of healing had, in this instance, amplified the illness rather than containing it. And the relationship between the community and the institution of Western medicine, which the Belgian mission represented, became for a period deeply complicated and uncertain. Families had watched their loved ones die in circumstances that did not permit the traditional mourning practices that gave death its proper social meaning in that culture. The close physical contact of washing and preparing the body, the gatherings of extended family and community, the rituals that transformed grief into shared communal experience. Because these very practices had been identified as vectors of transmission and had been, out of necessity, curtailed or forbidden by the public health response. The disruption of these rituals was, for many families and communities, a secondary wound alongside the primary wound of the illness itself, and it is a dimension of the Ebola story. This collision between the requirements of public health containment and the requirements of cultural and spiritual practice that would recur with notable consequence in every significant Ebola outbreak for the next 4 decades. Over the next several years, the institutional response to the 1976 outbreaks produced a modest but meaningful body of infrastructure for monitoring and responding to future Ebola events in Central Africa, though the resources devoted to this effort were, in retrospect, substantially smaller than the scale of the threat warranted. The World Health Organization maintained surveillance networks in the affected regions, and national health ministries in Zaire and Sudan developed, with varying degrees of effectiveness, protocols for recognizing and reporting cases of hemorrhagic fever that might represent Ebola or a related pathogen. The CDC in Atlanta maintained stocks of reagents and protocols necessary for laboratory confirmation of Ebola, and the Institute of Tropical Medicine in Antwerp continued its research into the epidemiology and biology of the virus. These were quiet, unglamorous, procedural forms of preparedness, the kind that do not attract attention or generate urgency in the way that an active outbreak does, but that represent the essential infrastructure through which societies attempt to translate the lessons of past disasters into the capacity to respond more effectively to future ones. It is worth noting that this infrastructure was assembled and maintained largely by people whose names do not appear in history books: laboratory technicians and field epidemiologists and public health administrators who did their work carefully and methodically, year after year, in the long quiet intervals between outbreaks, sustained by a professional commitment to a kind of vigilance that the world tends to notice only when it fails. The 1990s brought Ebola back into the human world with a frequency and a geographic distribution that suggested the virus was not the rare, isolated phenomenon that the relative quiet of the 1980s might have implied, but rather a recurring presence in the ecology of Central and Equatorial Africa, capable of emerging from its reservoir with unpredictable timing, and in communities that had no particular reason to expect it. In 1994, a French ethologist named Christophe Boesch, working in the Thai forest of Ivory Coast, found that chimpanzees in his study group were dying of what appeared to be hemorrhagic fever. And when one of his colleagues, a Swiss ethologist named Nathalie Molyneux, who had been working closely with the animals, fell ill shortly thereafter with symptoms consistent with the pattern identified in the 1976 outbreaks, laboratory investigation confirmed the presence of a new strain of Ebola, one that had not been identified before and which was subsequently named Ebola Thai forest. Molyneux was evacuated to Switzerland for treatment and survived, becoming one of the very few people ever to have contracted Ebola outside of Central Africa, and one of the very few confirmed survivors of the 1990s, period of the virus's resurgence. The Thai forest outbreak was small, involving only this single confirmed human case, but it demonstrated that the virus was present across a far wider geographic range in Africa than the 1976 data had implied, and that it was capable of crossing from wild primates to humans in the context of direct contact with infected animal tissues. A finding that pointed toward the complexity and the depth of the virus's relationship with the ecosystems of the African forest. In 1995, a more extensive outbreak occurred in the town of Kikwit in the Democratic Republic of Congo. Zaire had been renamed and would soon undergo further political transformation, bringing Ebola back to the same country where the first formally recognized outbreak had occurred nearly 20 years earlier. The Kikwit outbreak began in January 1995 and lasted until the end of July, producing 315 cases and 250 deaths, with a case fatality rate of approximately 79%, and with a pattern of transmission that was, in its essential structure, remarkably similar to the 1976 Yambuku outbreak. Centered initially on a hospital, spread by inadequate infection control practices, amplified by the movement of patients through the healthcare system, before the cause of the illness was recognized. The index case in Kikwit was a man who had been working in the forest outside the city, farming and charcoal making, in areas that brought him into regular contact with the forest ecosystem, and who fell ill in January 1995 and was admitted to Kikwit General Hospital, where the standard procedures of the time were insufficient to prevent transmission to other patients and to healthcare workers. By the time the cause of the outbreak was confirmed as Ebola in May 1995, confirmed through samples sent to the CDC in Atlanta, the virus had already circulated within the hospital system for several months, and the response required the same combination of isolation, contact tracing, and intensive infection control that had worked in Yambuku in 1976. But that was now being applied in a larger, more densely populated urban setting, with all the additional complexity that such a setting entails. The world's attention to the Kikwit outbreak of 1995 was notably greater than its attention to the 1976 events had been, in part because the global media environment had changed considerably over the intervening 2 decades, and in part because 2 events in 1994 and 1995 had brought viral hemorrhagic fevers into Western popular consciousness in a way that made news editors and their audiences more attentive to reports of outbreaks in Africa than they might previously have been. In 1994, Richard Preston had published a book titled The Hot Zone, which described in considerable detail the 1989 appearance of Ebola reston, a strain of the virus found in laboratory monkeys imported to the United States from the Philippines, and which, while focused on events that had not resulted in human illness, presented Ebola to a popular readership as a virus of extraordinary and almost mythological danger. In 1995, a film titled Outbreak, loosely inspired by the same constellation of fears, had been released to considerable commercial success, and the combination of Preston's book and the film had created in the Western public mind an image of Ebola that was vivid, frightening, and not entirely accurate in its details, but that served to ensure that when Kikwit began generating news, those reports reached audiences that had at least a general, if somewhat distorted, frame of reference for understanding what they were being told. The attention was not without consequence. International aid and expertise arrived in Kikwit more quickly than might otherwise have been the case, and the outbreak, though it was devastating for the community of Kikwit and for the healthcare workers who bore such a disproportionate share of its mortality, was contained within 7 months. The period between Kikwit in 1995 and the beginning of the 21st century saw additional outbreaks of Ebola in Uganda, Gabon, and the Republic of Congo, each one producing its own local history of illness and response, each one adding to the slowly accumulating scientific and epidemiological understanding of the virus, and each one confronting anew the persistent structural challenges: inadequate healthcare infrastructure, insufficient laboratory capacity, strained public health systems, and the complex social and cultural dynamics of communities navigating the dual catastrophe of epidemic disease and the public health measures designed to contain it. In Uganda in 2000 and 2001, an outbreak of Ebola Sudan in the Gulu district produced 425 cases and 224 deaths, making it the largest single Ebola outbreak recorded up to that point, and exposing once again the particular vulnerability of healthcare workers in settings where protective equipment is scarce and where the volume of patients makes strict adherence to infection control protocols exceptionally difficult. In Gabon and the Republic of Congo, outbreaks in the late 1990s and early 2000s were associated with handling the bodies of dead forest animals, particularly chimpanzees and gorillas, and added to the growing body of evidence linking spillover events to direct human contact with infected primate carcasses, though the question of the ultimate reservoir, the species that maintain the virus in the intervals between outbreaks, remained unanswered and the subject of ongoing, patient scientific inquiry. It is also during this period, in the late 1990s and the early years of the new century, that the first systematic investigations of fruit bats as a potential reservoir for Ebola began to produce results that pointed, with increasing consistency, toward this hypothesis. Researchers conducting surveillance studies in the forests of Central and West Africa found antibodies to Ebola virus in blood samples taken from several species of fruit bats, and in some samples found viral RNA sequences consistent with Ebola, suggesting that these animals were not simply occasionally exposed to the virus, but had sustained exposure to it over time, in a way consistent with a reservoir relationship. Fruit bats of the family Pteropodidae are, in the ecology of the African forest, animals of considerable ecological importance. They are pollinators and seed dispersers, and they range widely across the forest, roosting in large colonies, in trees and in caves, and coming into regular contact with humans through the harvesting of the forest trees, in which they roost, and through the direct hunting and consumption of bats as a food source, which is widespread across many parts of Equatorial and West Africa. The epidemiological significance of this finding was substantial because it meant that the risk of spillover events into human populations was not a rare and anomalous occurrence, but a structural feature of the relationship between human communities and the forest ecosystems in which they lived and worked. A risk that was, in a sense, built into the daily life of the region in ways that could not be simply abolished without fundamentally altering the way in which those communities related to their environment. The scientific community's evolving understanding of Ebola's ecology and biology during the 1990s and early 2000s was accompanied by the first serious attempts to develop medical countermeasures against the virus. Vaccines, antivirals, and therapeutic approaches that might reduce the catastrophic case fatality rates that had characterized every outbreak since 1976. This work proceeded slowly, as work on pathogens that can only be handled under biosafety level 4 conditions necessarily does, constrained by the specialized infrastructure required. The small number of institutions capable of maintaining such facilities, and the limited commercial incentive for pharmaceutical companies to invest in treatments for a disease that, however devastating in its local impact, affected relatively small numbers of people and was confined, at that point in its history, to remote areas of Central Africa. Government and academic research programs in the United States, Russia, Canada, and Germany led the primary research effort, and by the early 2000s, several candidate vaccines had shown promise in animal models, particularly in non-human primates, though the path from promising animal data to a tested and approved human vaccine is long, expensive, and procedurally demanding in ways that meant no vaccine was yet available when the next major outbreak came. The absence of a vaccine or an effective antiviral treatment was not for lack of scientific effort or scientific understanding. It was, rather, a consequence of the particular economic and institutional architecture through which the world produces medical interventions, an architecture that tended to prioritize conditions affecting large numbers of people in wealthy countries over conditions affecting smaller numbers of people in poor ones, even when those conditions carried fatality rates of the magnitude that Ebola consistently demonstrated. In the early years of the 21st century, as scientific research continued, and as the world's attention moved through its familiar cycle of engagement and disengagement with the ongoing story of Ebola in Central Africa, the virus itself was moving through the ecology of the African forest in ways that were not yet visible to the surveillance systems that existed at the time. It is worth allowing the mind to rest with this fact for a moment, to hold the image of a continent-wide forest, vast and complex and ancient, beyond any human reckoning, through which an invisible passenger moved in the blood of animals whose lives were largely unknown to science, persisting through the seasons and the decades with a patience and an indifference to human concern that no biological entity possesses by intent, but that every successful pathogen possesses by necessity. The forest did not know the names of the villages at its edges. The bats roosting in the great trees did not know the significance of what they carried. The children who played under those trees did not know, and could not have been expected to know, that the ecological relationship between their communities and the forest around them contained, within its ordinary and unhurried complexity, the conditions for something that the world would eventually be forced, in 2014, to notice on a global scale in a part of Africa, West Africa, where Ebola had never before been formally recorded, in a chain of events that would begin as all such chains begin, with a single moment that was small and unremarkable until, in retrospect, it was not. The Republic of Guinea, in the southeastern corner of the country near the borders with Sierra Leone and Liberia, is a region of remarkable biological diversity and human complexity, a landscape of forest and farmland, and small market towns connected by red dirt, roads, home to communities with deep traditions of cross-border mobility and trade that have persisted across generations, regardless of the political boundaries drawn by colonial administrations in the 19th and 20th centuries. It was in this landscape, in the village of Meliandou, in the Gekadou prefecture, that a child of approximately 18 months of age, a boy whose family knew him by the name Emile Wamuno, fell ill in December 2013 with fever and gastrointestinal symptoms that were, in their initial presentation, consistent with any of several common illnesses of childhood in that region. Emile died on the 28th of December 2013 and was followed in the subsequent days and weeks by his sister and his mother and his grandmother, 4 deaths in a single household 4 members of a family taken by an illness that moved through the family with the quiet and terrible efficiency that would, within months, define the character of what was coming. Epidemiologists who later reconstructed the chain of events from interviews with survivors and community members concluded that Emile had most likely been exposed to the virus through contact with bats roosting in a hollow tree near the family's home in Meliandou, a tree that children from the village reportedly played around regularly, and that had apparently harbored a colony of insectivorous bats of a species associated with Ebola viral sequences in prior studies. The hollow tree burned down at some point during the early months of 2014, before its epidemiological significance was fully understood, which means that the physical site of what would prove to be the index case of the largest Ebola outbreak in history no longer exists, lost to fire in the ordinary rhythms of village life. The illness that killed Emile Wamuno and his immediate family in late December 2013 and early January 2014 spread outward from Meliandou through the same mechanisms that had driven every Ebola outbreak before it. Through close contact between the ill and those who cared for them, through the mourning and burial practices that brought community members into physical contact with the bodies of the dead, and through the healthcare facilities to which the sick were brought for treatment, without, in those earliest weeks, any recognition of the pathogen that was responsible for their illness. The illness moved along the networks of family and community connection that tied the villages of the Gekadu prefecture to each other and to the larger market towns of southern Guinea, and it moved with the traders and the travellers and the mourners who attended funerals in multiple villages, carrying with them, unknowingly, the invisible passenger that had moved from the forest to Emile, and from Emile to his family, and from his family to the community, and from the community outward along the roads and the river paths of the forest borderlands. By February 2014, cases had appeared in the prefectural capital of Gekadu, and by March they had been reported in the capital city of Conakry, on the coast of Guinea, several hundred kilometers from Meliandou, a distance that spoke not to the biological capabilities of the virus, but to the human geography of a region where movement is continuous and constant, where families are scattered across borders, and where the social networks of mourning and commerce connect communities across distances that would, in a more sedentary world, have served as natural barriers to the spread of illness. It was not until late March 2014 that the World Health Organization received official notification from the Guinean Ministry of Health of an outbreak of hemorrhagic fever in the southeastern region of the country, and it was not until March 22nd, 2014, that laboratory testing, conducted partly at the Institut Pasteur in Lyon, France, and confirmed at the Institut Pasteur in Dakar, Senegal, confirmed the cause of the outbreak as Ebola virus Zaire, the most lethal of the known Ebola strains, the same strain that had been responsible for the 1976 Yambuku outbreak and the 1995 Kikwit outbreak, and most of the major Central African outbreaks in the intervening decades. The WHO announced the outbreak publicly on March 23, 2014, reporting 49 confirmed cases and 29 deaths in Guinea, a case fatality rate of approximately 59%, severe and deeply sorrowful, but within the range of what previous Ebola Zaire outbreaks had produced. The announcement was received in the initial days of its circulation with something less than the alarm that the full scope of what was coming would eventually warrant. The global health community had responded to Ebola outbreaks before. The protocols were established. The institutions were prepared. There was, in those first weeks of official recognition, a reasonable, if ultimately incorrect, assumption that the Guinean outbreak would follow the pattern of its predecessors, that it would be contained through isolation and quarantine and community engagement within a period of months, leaving behind it a community transformed by loss, but an outbreak ultimately controlled. The forest, however, had arranged things differently this time, and the months that followed would demonstrate, with quiet and accumulating force, exactly how different this particular emergence of the virus was from anything that had come before it. The weeks that followed the WHO's announcement of March 23, 2014, were weeks of measured and professional response. The mobilization of field teams, the deployment of experts, the activation of the protocols that had been developed and refined through decades of experience with Ebola in Central Africa. And yet they were also weeks in which something was happening in the forest borderlands of southeastern Guinea that those protocols had not been designed to address, something that had to do not with the biology of the virus, but with the particular human geography of the region in which it had chosen, if one may use so intentional a word for so unconscious a process, to emerge. The Gekadu prefecture sits at the intersection of three national borders: Guinea, Sierra Leone, and Liberia, and the communities that live along those borders have, for generations, organized their social and economic lives without particular reference to the lines that colonial mapmakers drew across the forest in the 19th century. Families have members in all three countries. Markets draw traders from across all three borders. Funerals are attended by relatives who may travel many hours across multiple borders to pay their respects and to participate in the mourning, ceremonies that are, in the cultures of this region, among the most socially and spiritually significant events in communal life. Healthcare, where it exists, is sought wherever it can be found. Across the border in Sierra Leone, if the Guinean clinic is too far, in Liberia, if a relative lives there and can provide accommodation during treatment, the movement of people through this landscape is not anomalous or unusual. It is the fundamental social fabric of the region, woven over centuries of shared history and common language and intermarriage and trade. And it is precisely this fabric, this richness of human connection, that would allow the virus to move in ways that no previous Ebola outbreak had moved, crossing borders not through any dramatic act, but through the ordinary daily practice of being a community. By April 2014, cases of Ebola had been confirmed in Liberia, appearing in the county of Lofa, near the border with Guinea, brought there by individuals who had attended funerals in the Guinean border region and had returned home carrying the virus without knowing it. The Liberian Ministry of Health notified the WHO, and response teams were deployed, and the same procedures that had been initiated in Guinea were initiated in Liberia. Contact tracing, isolation of confirmed cases, community engagement to explain the nature of the illness, and the reasons for the public health measures being recommended. In May 2014, the first cases appeared in Sierra Leone, again near the border with Guinea, again connected through the social networks of mourning and family connection that linked the communities of the forest borderlands across the lines that the maps insisted upon, and that the communities themselves treated as a secondary consideration at best. The three countries were responding to their outbreaks with the resources available to them, and those resources were, it is important to acknowledge plainly, severely limited. Limited in the number of trained healthcare workers, in the quantity of personal protective equipment, in the number of functioning laboratory facilities capable of confirming Ebola diagnoses, in the capacity of the health ministries to coordinate complex multi-agency responses across large geographic areas with poor road infrastructure and unreliable communications. These were countries that had emerged from devastating civil conflicts. Sierra Leone's civil war had ended in 2002, Liberia's in 2003, and whose healthcare systems had been profoundly damaged by years of conflict, displacement, and underfunding, and which were in the early and difficult stages of reconstruction when the outbreak arrived. The virus had found, through no design of its own, a set of circumstances that would allow it to do something it had never done in any previous outbreak, in any previous location, to sustain itself in large human populations for an extended period without being contained. During this period, from April through June 2014, the international response remained calibrated to the assumption that the outbreak was following a pattern consistent with previous Ebola events, severe, yes, and requiring significant effort to control. But ultimately, the kind of outbreak that had been controlled before and could be controlled again through the application of known methods by experienced teams. Médecins Sans Frontières, the international medical humanitarian organization known in the English-speaking world as Doctors Without Borders, had been present in the region since the earliest days of the outbreak and had been sounding a note of increasingly urgent concern that the scale and the geographic distribution of the outbreak exceeded what the current response was equipped to address. In a statement issued in June 2014, MSF described the outbreak as out of control and called for a dramatically expanded response. More treatment centres, more staff, more resources, a characterization that was at the time viewed by some in the global health establishment as alarmist, but that would, within weeks, be recognized as an accurate description of a situation that had been developing through the spring of 2014 without receiving the level of attention and resource that it required. The international health community was not indifferent. It was, rather, working within the institutional frameworks and the assumptions that its prior experience with Ebola had established, and those frameworks and assumptions had been built around outbreaks that were smaller, more geographically concentrated, and occurring in countries with very different histories and very different social structures than the three countries of the Mano River region. The mismatch between the established framework and the emerging reality was not immediately visible from inside the framework, which is one of the most fundamental and recurring challenges of public health emergency response. The difficulty of recognizing, in time, that a new event is categorically different from the events that one's protocols were designed to address. The month of June 2014 was, in retrospect, a month of crucial and tragic significance in the trajectory of the West African Ebola epidemic. A month in which the case counts were climbing, and the geographic spread was expanding, and the healthcare systems of Guinea, Liberia, and Sierra Leone were beginning to show the early signs of the strain that would, in subsequent months, bring them close to complete collapse. In Sierra Leone, an event occurred in late May and early June that accelerated the epidemic in that country in ways that would not be fully understood until epidemiologists later reconstructed the chain of transmission with the careful patience of historical investigation. A traditional healer in the Kailahun district of Sierra Leone, a woman of considerable local reputation who had been treating patients with symptoms of the illness using the healing methods of her tradition, died of Ebola herself in late May, and her funeral was attended by large numbers of mourners from across the district and from neighboring areas of Guinea, many of whom participated in the mourning and burial rituals that, in the culture of this region, involve close physical contact with the body of the deceased. The funeral became, in epidemiological terms, what is known as a super-spreading event, a single occasion from which dozens of secondary transmission chains radiated outward across a wide geographic area, seeding cases in multiple locations simultaneously, and making the subsequent work of contact tracing extraordinarily difficult because the network of connections was suddenly so much larger and more complex than it had been the day before. This pattern, the funeral as a vector of transmission, the mourning ceremony as a mechanism of spread, would recur repeatedly through the West African epidemic, and it would force the public health response to grapple over and over again with the profound tension between the epidemiological necessity of preventing contact with the bodies of the deceased and the spiritual and cultural necessity, as understood by the affected communities, of honoring the dead in the manner that their traditions required. The response to this tension between the requirements of infection control and the requirements of cultural and spiritual practice was one of the most important and most difficult aspects of the entire West African epidemic response, and it is worth dwelling on it with the care and the thoughtfulness that it deserves, because it touches on questions that go far beyond the specifics of Ebola, and that bear on the fundamental relationship between public health, authority, and community autonomy in contexts of epidemic disease. The communities of Guinea, Sierra Leone, and Liberia were not, it is deeply important to understand, simply refusing to comply with public health guidance out of ignorance or irrationality or indifference to their own safety. They were navigating an extraordinarily difficult situation in which the instructions they were receiving from health authorities: do not touch the bodies of the dead, do not gather for funerals, do not care for sick relatives in the traditional manner, required them to abandon practices that were, in their understanding of the world, not merely cultural preferences, but moral and spiritual obligations, obligations to their ancestors and to their community, and to the proper ordering of the relationship between the living and the dead. To be told that one cannot prepare the body of one's mother, cannot wash and dress her, and sit with her through the night before burial, is to be told not merely that one must change a habit, but that one must fail in a fundamental duty. And the weight of that failure, the grief and the shame and the spiritual anxiety that it produces, is not something that a public health notice or an instruction from a government official can easily address or adequately compensate. The most effective responses to this challenge in the West African epidemic were those that worked collaboratively with community leaders and traditional healers and religious authorities to develop modified burial practices that preserved as much as possible of the cultural and spiritual content of traditional mourning, while eliminating the specific practices that posed the greatest transmission risk, an approach that required time and patience and genuine respect for the community's own knowledge and values. And that produced, where it was implemented well, significantly better community cooperation with the overall response. By July 2014, the epidemic had reached the capital cities of all three most affected countries: Conakry in Guinea, Monrovia in Liberia, and Freetown in Sierra Leone. And the presence of the virus in urban settings with populations in the hundreds of thousands represented a qualitative shift in the nature of the outbreak that the global health community was beginning, with a mixture of alarm and urgent mobilization to absorb. Ebola in a remote rural setting is, from an epidemiological perspective, a very different kind of problem from Ebola in a dense urban environment, where healthcare facilities are overwhelmed, where the movement of people is constant and difficult to monitor, and where the social networks along which the virus can travel are orders of magnitude larger and more complex than in the village settings, where every previous outbreak had been contained. Monrovia, the capital of Liberia, had a population of approximately 1 million people at the time, and in July and August of 2014, it became the epicenter of the most intense urban Ebola transmission ever recorded, with cases appearing in poor and densely populated neighborhoods where healthcare access was minimal, where understanding of the illness and its transmission was incomplete, and where the fear generated by the outbreak interacted with the trauma of Liberia's recent civil war history in ways that made community engagement and trust building exceptionally complex and demanding work. The healthcare system of Liberia, which had been one of the weakest in the world, even before the outbreak, was being overwhelmed, not only by Ebola cases, but by the secondary effects of the epidemic on all other healthcare, as clinics closed and healthcare workers fell ill or stayed home out of fear, and patients with malaria and tuberculosis and obstetric emergencies and childhood illnesses found no care available because the system had been consumed by a single catastrophic demand. It was in this context, the context of 3 capitals affected, healthcare systems collapsing, case counts rising exponentially, and the global health response still calibrated to a smaller and more manageable kind of outbreak, that a single event in late July 2014 fundamentally changed the tenor of the international conversation about the West African epidemic. A Liberian-American man named Patrick Sawyer, a consultant who had been working for the Liberian government, fell ill in Monrovia in late July after having attended the funeral of his sister, who had died of Ebola, and traveled by air to Lagos, Nigeria, where he collapsed at the airport on July 20, 2014, and was taken to a private hospital. Sawyer died on July 25, 2014, becoming the first Ebola fatality on Nigerian soil, and triggering a response in Africa's most populous country that would, in the subsequent weeks, demonstrate both the terrible vulnerability of the disease to introduction through international air travel and the extraordinary capacity of a well-resourced and well-organized public health response to contain it. Nigeria's response to the Lagos outbreak has been widely studied and widely praised in the subsequent years. The rapid identification and isolation of contacts, the aggressive contact tracing, the strong leadership of a dedicated response team, and the existence of an emergency operations center that had been established with support from the CDC in the years following the polio eradication campaign. And it succeeded in containing the Lagos outbreak with 19 confirmed cases and 8 deaths before it could spread into the enormous and densely populated urban fabric of a city of 20 million people. But the significance of Sawyer's arrival in Lagos extended beyond the outbreak it caused, because it demonstrated to the world, with sudden and clarifying force, that the West African epidemic was not a contained regional crisis, but a potential global one, and that the long-established mechanisms of international health governance were being tested in ways that they had not previously been required to address. The international response to the West African Ebola epidemic accelerated dramatically in August 2014 driven by a combination of the exponentially rising case counts in Guinea, Liberia, and Sierra Leone, the arrival of Ebola in Nigeria, and the subsequent identification of cases in Senegal and Mali, and the infections of several Western healthcare workers who had been treating Ebola patients in West Africa and who were evacuated to hospitals in the United States and Europe for treatment. The evacuation of these Western patients, including Dr. Kent Brantley and nurse Nancy Wrightball, both Americans working with the faith-based organization, Samaritan's Purse in Liberia, who were treated at Emory University Hospital in Atlanta, beginning in August 2014, brought the epidemic into the living rooms and news feeds of Western audiences in a way that the deaths of tens of thousands of West Africans had not. And this shift in visibility, however uncomfortable it may be to acknowledge, had real consequences for the political will and the financial resources that began to flow toward the response in the weeks that followed. On August 2014, the World Health Organization declared the West African Ebola outbreak a public health emergency of international concern, the highest level of alert available to the organization under the international health regulations, a BISPI declaration that triggered the obligations of WHO member states to coordinate their responses and to make resources available. And that represented, in formal and institutional terms, the acknowledgement this was no longer a regional outbreak, but a global crisis requiring a global response. The weeks that followed the FAIC Declaration of August 8, 2014 were weeks of extraordinary activity in the corridors of international health, governance, meetings at the United Nations and at the WHO, emergency sessions of national governments, the mobilization of military and civilian resources from the United States, the United Kingdom, France, and other nations, and the beginning of a massive scale-up in the international response that included the construction of Ebola treatment units, the deployment of thousands of international workers, and the commitment of billions of dollars in emergency funding. The United States, under President Barack Obama, announced a major military-led response in mid-September 2014, committing 3,000 American military personnel to Liberia to support the construction of treatment facilities and the training of healthcare workers, a deployment that was unprecedented in its scale and in its framing of a disease outbreak as a matter of national security and international strategic concern. The United Kingdom took the lead on the response in Sierra Leone, and France on the response in Guinea, with each country bringing to its designated area not only military and technical resources, but the particular relationships and institutional connections that grew from their historical ties to these former colonies, relationships that were complicated by the weight of colonial history, but that were also, in practical terms, meaningful networks of trust and communication that the response could build upon. The UN Secretary-General Ban Ki-moon established the UN Mission for Ebola Emergency Response, known as UNMIR, in September 2014, the first ever UN Emergency Health Mission, a reflection of the recognition that the response to this epidemic required a level of coordination and resource that exceeded the capacity of any single agency or organization. Meanwhile, in the affected communities of Guinea, Liberia, and Sierra Leone, the scale of the epidemic in the late summer and autumn of 2014 produced a quality of experience for which the historical record does not have adequate words, not because it was beyond description, but because the descriptions that exist in the testimonies of survivors and healthcare workers and community members are so deeply particular and so deeply human that any summary of them risks flattening the individual weight of each account into the abstraction of statistics and trends. Healthcare workers in Liberia worked in improvised treatment facilities, overwhelmed with patients, wearing personal protective equipment in tropical heat for hours at a time, watching colleagues fall ill and die, continuing to come to work in circumstances that required a quality of courage that those of us who have not faced it are perhaps not fully equipped to measure. Community health workers walked through neighborhoods distributing information and listening to fear and grief and anger, and doing so without the protection that their more formally trained colleagues had because the epidemic was too large and the formally trained workers too few. Families separated themselves from sick relatives with grief and with guilt, following instructions that they understood to be necessary, but that cost them something that no public health framework can quantify. Children were orphaned in numbers that would eventually require a separate, dedicated response. By the end of the epidemic, an estimated 17,000 children in the 3 most affected countries had lost one or both parents to Ebola, and the social and psychological consequences of this loss were layered on top of societies already bearing the accumulated trauma of years of civil conflict. It is worth allowing this human dimension of the epidemic to be present in the narrative, not because it adds urgency or drama. We are not seeking urgency or drama here, but because the epidemic was, at its most essential level, not a phenomenon of virology or epidemiology, but a human experience, lived by specific people in specific places, people who had names and families and histories and futures that were altered or ended by an event that no one had chosen and no one had been adequately prepared to face. The Ebola treatment units that began to be constructed in Liberia in September and October 2014, with the support of the American military engineering teams that had been deployed to the country, represented one of the most significant practical contributions of the international response to the management of the epidemic. Previous Ebola treatment facilities in West Africa had been overwhelmed by the volume of patients, and one of the most dangerous consequences of this overwhelm was that many patients who presented at treatment facilities had to wait, sometimes for days, in conditions of inadequate isolation, in close proximity to other patients and to family members and to healthcare workers who had not yet received the confirmation that the person in front of them was Ebola positive, and who were therefore managing the tension between the imperative to provide care and the imperative to protect themselves from a virus, for which the available protective equipment was already in desperately short supply. The new treatment centers, constructed with American military support, were designed with a capacity of 100 beds each, with physical separation between suspected, probable, and confirmed cases, with dedicated pathways for the movement of patients and staff that minimized the risk of cross-contamination, and with improved systems for waste management, for the safe burial of those who died within the facility, and for the psychological support of the healthcare workers who staffed them. The construction of these facilities did not immediately solve the epidemic, because the epidemic was far too large and far too geographically distributed for any single intervention to address. But it represented a meaningful addition to the capacity of the response, and it demonstrated, in the particular way, that large physical structures always demonstrate the intentions of those who build them, that the international community was attempting, with whatever resources it could mobilize, to respond to the scale of what was happening. The arc of the epidemic in Liberia during the autumn of 2014 followed a pattern that epidemiologists would later study with considerable interest, because the peak of transmission in Liberia appears to have occurred in September 2014, before the full scale of the international response had arrived and before the major new treatment facilities had been constructed, suggesting that changes in community behavior, driven by the growing fear and awareness within the community itself, played a significant role in reducing transmission, independently of the formal international response. Communities in Monrovia and elsewhere began to change their burial practices, to isolate the sick more rigorously, and to reduce the social gatherings that had served as amplification events for the virus, not primarily because they were instructed to do so by public health authorities, but because the epidemic had become visible enough and terrible enough that the need for change was being understood from the inside of the community rather than being imposed from the outside. This observation, that community behavior change, driven by community knowledge and community decision-making, was a crucial element of epidemic control, would become one of the most important lessons of the West African epidemic for the field of global health, reinforcing a body of evidence from HIV, cholera, and other epidemic diseases, suggesting that top-down, authority-driven public health responses are consistently less effective than responses that are built in genuine partnership with the communities they are designed to serve. The communities of West Africa were not passive recipients of international assistance. They were active participants in the management of their own crisis, and their knowledge of their own social structures, their own cultural practices, and their own networks of trust and influence was, in many instances, more practically valuable to the response than the protocols developed in offices in Geneva or Atlanta. Sierra Leone's epidemic followed a somewhat different temporal pattern from Liberia's, with the peak of transmission occurring somewhat later, in October and November 2014, partly because the outbreak in Sierra Leone had accelerated later than in Liberia, and partly because certain aspects of the response in Sierra Leone took longer to scale up to the required level. The government of Sierra Leone, working with the UK military and with international NGOs, including MSF and the International Federation of Red Cross and Red Crescent Societies, implemented a series of measures that were, in some respects, among the most intensive ever applied to an Ebola outbreak, including, controversially, a series of 3-day nationwide lockdowns in September, October, and November 2014, during which the entire population of the country was asked to remain at home while community health workers moved through every neighborhood and every village, conducting house-to-house surveillance and identifying sick individuals for testing. These lockdowns, known as the Western Area Surge in Freetown, were logistically extraordinary undertakings, requiring the coordination of thousands of community health workers, the suspension of all normal economic activity for their duration, and the cooperation of a population that was already under enormous stress. Their epidemiological impact was debated within the public health community. Some analyses suggested they accelerated the decline in transmission, others were less certain, but they represented, regardless of their precise effectiveness, a reflection of the determination of the Sierra Leonean government and its partners to deploy every available tool in the effort to end the epidemic. In Guinea, the epidemic followed yet another pattern, one that was in some ways the most challenging of the three, because the outbreak in Guinea was, relative to Liberia and Sierra Leone, more geographically dispersed, more persistent in remote forest areas that were difficult to access, and where Community engagement was particularly challenging and more prone to a pattern of apparent decline followed by resurgence that epidemiologists describe as a flare-up pattern. Small clusters of new cases appearing in areas where transmission had seemed to have been interrupted, often connected to a single funeral or a single healthcare facility, spreading briefly before being contained, then appearing again somewhere else along the network of forest communities that connected the southeastern prefectures of Guinea. The Guinea response faced challenges specific to that country's political and social context, including a degree of community resistance in certain areas that was, in part, a product of the particular history of the relationship between the Guinean state and its rural communities. A relationship marked by significant mutual suspicion arising from decades of authoritarian governance and the experience of state violence that many communities in the affected regions carried in their recent memory. When health authorities arrived in white vehicles, wearing unfamiliar protective equipment, and began removing sick community members to treatment facilities, from which some of them did not return, the response in some communities was not cooperation, but fear. Fear that the authorities were doing harm rather than good, that the treatment facilities were places of death rather than healing, that the disease itself was in some way connected to the presence of foreign workers and foreign organizations in their communities. In September 2014, in the prefecture of Zaire in southeastern Guinea, a delegation of healthcare workers and local officials conducting community sensitization was attacked and killed by villagers who feared that the team was spreading the disease rather than attempting to contain it. A devastating event that spoke to the depth of the trust deficit between the public health response and the communities it was trying to serve, and that underscored with terrible clarity the consequences of that deficit for the people on both sides of it. The international scientific community's engagement with the West African epidemic was not limited to the practical work of response. The construction of treatment facilities and the deployment of contact tracers and the training of healthcare workers, but extended to an extraordinary mobilization of research capacity that produced, during the course of the epidemic, a volume and a velocity of scientific publication that had never previously been associated with an infectious disease outbreak of this kind. Researchers from universities and research institutes in the United States, the United Kingdom, France, Germany, Canada, and the affected countries themselves conducted epidemiological analyses, phylogenetic studies of the viral genome, clinical trials of candidate therapeutics, and ethnographic investigations of community responses that together constituted one of the most intensive and most productive scientific investigations ever mounted in real time during an ongoing epidemic. One of the most significant of these scientific contributions was a genomic sequencing study published in the journal Science in August 2014, led by researchers from the Broad Institute of MIT and Harvard, in collaboration with scientists from Sierra Leone's Kenema Government Hospital, which sequenced the genomes of 99 Ebola virus samples collected from 78 patients in Sierra Leone, and used this data to reconstruct the evolutionary history of the virus as it moved through the human population. The study, which acknowledged the deaths of 5 of its Sierra Leonean co-authors from Ebola before its publication, found that the West African epidemic had been seeded by a single introduction of the virus from the animal reservoir into the human population, the index case in Meliandou, and had then spread and diversified within the human population through sustained human-to-human transmission, accumulating genetic mutations at a rate that was consistent with other RNA viruses, and that had, by the time of the study, produced a viral population in West Africa that was genetically distinct from the Ebola strains that had caused outbreaks in Central Africa. The acknowledgement of the 5 deceased co-authors in the published paper was not a formality. It was a recognition that the science was being produced in circumstances where the scientists themselves were at risk, and that the knowledge gained from the study carried within it the weight of the lives that had been given in the course of its production. The clinical management of Ebola patients in the West African epidemic represented one of the most demanding and most instructive challenges in the history of modern medicine, not because the disease was biologically unprecedented. Its essential characteristics had been understood, at least in broad outline, since 1976. But because the scale at which it was now occurring required the practice of medicine under conditions that stripped away many of the resources and supports that modern clinical practice depends upon, and demanded instead a kind of resourceful, adaptive, fundamentally human engagement with very sick people in circumstances of extraordinary difficulty. The Ebola treatment units that had been established across the 3 most affected countries were places of structured protocol and careful procedure. The donning and doffing of personal protective equipment followed a precise sequence developed over decades of biosafety research, each step observed and checked by a colleague to reduce the risk of self-contamination in the moments of greatest fatigue and inattention. And yet, within that structure of protocol, the clinicians and nurses who staffed the units were working with a patient population whose needs went far beyond what the protocols could fully address. The primary medical intervention available to Ebola patients in the early months of the epidemic was supportive care, the replacement of fluids lost through the illness, the management of fever and discomfort, the monitoring of organ function, and the response to whatever complications the individual patient presented, and the delivery of even this relatively simple level of care was made enormously difficult by the protective equipment that had to be worn, which limited manual dexterity, restricted communication, and generated intense physical discomfort in the tropical heat, limiting the time any individual healthcare worker could spend at the bedside before needing to exit the treatment area to rehydrate and recover. There were experimental therapeutics available in small quantities, most notably a drug called ZMapp, a combination of 3 monoclonal antibodies that had shown promise in non-human primate studies, but had never been tested in humans. And the question of how to allocate these experimental treatments in a situation of extreme scarcity was one of the most ethically fraught dimensions of the epidemic response, generating extensive discussion within the global bioethics community about the obligations of the international medical establishment to the populations most affected by the disease, and about the conditions under which experimental treatments of unproven safety may ethically be offered to desperately ill patients. The experience of the healthcare workers who served in the West African epidemic, both international volunteers who came from outside the region, and, far more numerously, the local nurses and doctors and community health workers who remained in their own communities and continued to work through months and years of epidemic, constitutes one of the most important human threads in the entire story of Ebola, a thread that deserves to be followed with patience and with care, rather than reduced to statistics about healthcare worker infections and deaths. the healthcare workers of Guinea, Liberia, and Sierra Leone faced a situation that placed them in an impossible position. They had chosen, through their training and their professional commitment, to care for the sick, and the epidemic presented them with sick people in need of care, but the specific disease afflicting those people was one for which the act of caregiving itself carried a profound personal risk that was, in the early months of the epidemic, inadequately supported by the protective resources that should have been available. Healthcare worker infections became, during the epidemic, one of the most closely tracked and most deeply concerning metrics of the response. Not only because each infected healthcare worker was an individual human tragedy, but because the loss of healthcare workers to the epidemic reduced the capacity of the healthcare systems to respond to it in a self-reinforcing cycle that was one of the most dangerous structural features of the entire crisis. By the time the epidemic was effectively over, more than 800 healthcare workers had been infected with Ebola across Guinea, Liberia, and Sierra Leone, and approximately 500 of them had died. A loss that represented, in countries where the ratio of trained healthcare workers to population was already among the lowest in the world, a devastating blow to the very infrastructure that would be needed for the years of recovery and rebuilding that lay ahead. The names of these healthcare workers, the doctors and nurses, and laboratory technicians and ambulance drivers and burial team members, were recorded where the records permitted, and in many cases memorialized in the communities where they had served. And the weight of their sacrifice is not something that a documentary script or any other form of words can fully contain or adequately honor. Among the figures who emerged from the West African epidemic as emblems of the courage and the dedication of the whole healthcare workforce, a Liberian nurse named Salome Kawa, who contracted Ebola while caring for her parents, both of whom died, and who survived the illness and returned to work in an Ebola treatment facility, where her status as a survivor, and therefore as a person who carried some degree of immunity to reinfection, made her uniquely valuable as a caregiver for patients who needed close physical contact that other healthcare workers could not safely provide. Kawa was named, along with 3 other healthcare workers, as one of Time magazine's Persons of the Year for 2014, a recognition that brought her story to an international audience, and that served, in its small way, as an acknowledgement of the extraordinary human effort that was being made by ordinary people in extraordinary circumstances throughout the affected region. There were thousands of others like her, healthcare workers whose stories were not covered by international media, whose names did not appear in any magazine, but who made the same choices and bore the same risks, and demonstrated the same quality of commitment in the daily work of the epidemic response. There was, in the community of Ebola survivors in West Africa, a kind of solidarity and a kind of knowledge that could not be transferred by training or by protocol. The knowledge of having been inside the illness and come out the other side, the solidarity of shared experience in the face of a disease that was isolating in ways that went far beyond the physical isolation of the treatment unit. Because the stigma associated with Ebola survival in the affected communities meant that many survivors returned home to find themselves feared and avoided by the neighbors, and sometimes even the family members from whom they most needed support and welcome. The stigma of Ebola survival was one of the dimensions of the epidemic that received, in the early months of the international response, considerably less attention than the clinical and epidemiological dimensions and yet it was a dimension with profound consequences for individual survivors and for the broader dynamics of community response to the epidemic. In the cultures of West Africa, as in many cultures around the world, serious illness is understood within frameworks of meaning that extend beyond the biomedical. Illness may be understood as a consequence of spiritual transgression, of conflict with ancestors, of the action of malevolent forces, or of the breaking of social taboos. And Ebola, with its rapid progression, its high fatality rate, and its distinctive clinical presentation, generated in the communities. It touched a quality of fear and of interpretive anxiety that made it particularly susceptible to the attachment of stigmatizing meanings. Survivors in all three countries reported being rejected by their communities upon return home, being told to leave, having their possessions destroyed, being denied access to water and markets and social gatherings, being treated as carriers of contamination even months after their recovery and their formal medical clearance. Children who had survived Ebola were in some cases excluded from school. Adults were in some cases unable to return to their livelihoods. Women who had survived faced particular forms of stigma, rooted in the complex intersection of disease, gender, and social expectation. The psychosocial support programs that were eventually developed in collaboration with survivor communities. Programs that involved survivors, themselves as peer counselors and community educators, that worked with religious leaders and community elders to reframe the meaning of survival, and that provided practical support to survivors, navigating the difficult process of reintegration, represented one of the more thoughtful and more genuinely community-centered elements of the overall response. And their development and implementation constituted a meaningful contribution to the understanding of how epidemic response can be made more humane and more effective by taking seriously the social and psychological dimensions of disease and its aftermath. The trajectory of the epidemic through the winter of 2014 and into 2015 followed a pattern of gradual, uneven, geographically variable decline that reflected both the effects of the scaled-up response and the complex social and ecological dynamics of the affected region. In Liberia, the case counts that had been rising exponentially through September and October 2014 began to fall in November, and by January 2015, the country was reporting dramatically fewer new cases per week than it had been reporting at the peak of the epidemic, a decline that was celebrated cautiously by the response community, with the caution reflecting an awareness that previous apparent declines in the epidemic had been followed by resurgences, and that the declaration of victory was, in an epidemic of this complexity, always premature until the conditions for resurgence had been sufficiently understood and addressed. In Sierra Leone, the decline was slower and occurred somewhat later, with the country still reporting significant numbers of new cases through January and February 2001, 5 before the trend began to move consistently downward. In Guinea, as had been the pattern throughout the epidemic, the decline was the most fitful and the most prone to interruption by new clusters of cases in forest communities that had seemed, briefly, to have escaped the epidemic altogether, and then found themselves at its center once more, their remoteness having protected them from early exposure, but also having kept them outside the networks of information and support, through which the more accessible communities had been reached by the response. The three countries were, in a sense, experiencing three slightly different versions of the same epidemic, shaped by the same virus, but contoured by the particular geography demography, history, and institutional capacity of each nation, and requiring from the response a degree of flexibility and local adaptation that was not always easy to maintain within the frameworks of large international emergency operations that are, by their nature, oriented towards standardization and scalability. The question of experimental therapeutics and vaccine development, which had been present in the background of the epidemic since its earliest months, moved to the foreground of the international scientific response in late 2014 and into 2015 as the scale of the epidemic created both the ethical justification and the practical opportunity for clinical trials that would not have been possible in the context of the smaller, more rapidly contained outbreaks that had characterized Ebola's prior history. 3 candidate vaccines had been in development at the time of the epidemic's emergence: a vaccine developed by the Public Health Agency of Canada and licensed to Merck, a vaccine developed by Johnson & Johnson in collaboration with Bavarian Nordic, and a vaccine developed by GlaxoSmithKline in collaboration with the National Institute of Allergy and Infectious Diseases in the United States. The Canadian Merck vaccine, known as RVSV-Zebov, and subsequently commercialized under the name Ervebo, was the furthest along in development and became the subject of a landmark clinical trial conducted in Guinea in 2015. A trial that used an innovative design known as a ring vaccination strategy, in which the contacts of confirmed Ebola cases were randomized to receive the vaccine either immediately or with a delay of 3 weeks, allowing the comparison of infection rates between the two groups to provide an estimate of vaccine efficacy. The results of this trial, published in The Lancet in December 2015, found that among the approximately 4,000 individuals who received the vaccine immediately, there were 0 cases of Ebola infection beginning more than 10 days after vaccination, compared to 16 cases in the group that received the vaccine with a delay. A finding that, while based on a relatively small number of events and requiring cautious interpretation, suggested that the vaccine was highly effective and represented the culmination of nearly 4 decades of scientific effort to develop a medical countermeasure against the pathogen that had first been formally identified in a Belgian mission hospital in northern Zaire in 1976. The declaration of the end of the West African Ebola epidemic was made in stages by the World Health Organization over the course of 2015 and 2016. Liberia was declared Ebola-free for the first time on May 9, 2015, then experienced 2 small resurgences before being declared free of transmission for the final time in January 2016. Sierra Leone was declared free of Ebola transmission on November 7, 2015, and Guinea on December 29, 2015, though a small cluster of cases appeared in Guinea in March 2016, linked to the persistence of the virus in the semen of a male survivor, a finding that had emerged during the epidemic as one of the more unexpected and more troubling aspects of Ebola biology. The discovery that the virus could persist in immune-privileged sites in the body, including the testes, the eyes, and the central nervous system, for months after a person had recovered from the acute illness and cleared the virus from the bloodstream, with the potential for sexual transmission from recovering male survivors to their partners long after they had been formally cleared and discharged from treatment. The WHO declared the end of the public health emergency of international concern on March 29, 2016, and in June 2016, declared the end of all Ebola transmission in West Africa. concluding a 2.5-year epidemic that had produced, in its final accounting, 28,616 confirmed, probable, and suspected cases, and 11,310 deaths across 10 countries, with the vast majority of cases and deaths concentrated in Guinea, Liberia, and Sierra Leone. These numbers, vast as they are, represent only those cases and deaths that were recorded in a system under enormous strain, and epidemiologists have estimated that the true number of infections may have been considerably higher than the official count, since in the chaos of an overwhelmed healthcare system, many cases were never confirmed, many deaths were never reported, and the full human toll of the epidemic will never be known with the precision that the official statistics might seem to imply. Beyond the immediate numbers of cases and deaths, the West African Ebola epidemic left behind it a set of secondary consequences that would shape the health and the social fabric of the three most affected countries for years and decades to come. Consequences that are, perhaps, less dramatic than the epidemic itself, but no less real in their weight and their duration. The routine healthcare systems of Guinea, Liberia, and Sierra Leone had been severely disrupted during the epidemic period, and the disruption had consequences that extended far beyond Ebola. Disruption of vaccination campaigns meant that measles and other preventable diseases increased in the years following the epidemic, and the decline in health-seeking behavior among communities who had come to fear healthcare facilities as places associated with Ebola infection led to increased mortality from malaria, tuberculosis, HIV, and other conditions that are manageable when patients seek care early, and deadly when they do not. Studies conducted in the years following the epidemic found significant increases in maternal and neonatal mortality associated with the collapse of obstetric services during the epidemic period, women delivering at home without skilled birth attendants, women with obstetric complications unable to access surgical care, newborns dying from infections that would have been treatable in a functioning healthcare system. The economic consequences of the epidemic were also severe. The World Bank estimated in 2015 that the three most affected countries had suffered a combined economic loss of approximately $2.2 billion as a result of the epidemic, driven by the collapse of agriculture, trade, and investment during the period of the outbreak, and by the long-term costs of rebuilding healthcare and social systems. These consequences settled over the affected communities with the quiet weight of things that do not make headlines, but that shape the texture of daily life for years and years, and they are part of the full accounting of what the epidemic cost. An accounting that cannot be completed in the language of outbreak statistics, but requires the longer and more patient language of social and economic history. The global health governance lessons of the West African Ebola epidemic were debated with considerable urgency in the months and years following the end of the outbreak in reports and commissions and academic analyses that were produced by the WHO, the World Bank, the United Nations, national governments, and independent research institutions. And that reached, with broad though not universal consensus, a set of conclusions about the structural failures that had allowed a containable outbreak to become the largest Ebola epidemic in history. The most fundamental of these conclusions was that the international health community had responded too slowly and with too few resources in the critical months of early 2014, when the epidemic was still at a scale at which aggressive response might have contained it, and that this slow response was a product not of incompetence or indifference, but of structural features of the international health system. the underfunding of the WHO and other international health bodies, the absence of a dedicated global emergencies health fund, with the capacity to deploy resources quickly without the lengthy procurement and approval processes that govern normal international spending, the weakness of health surveillance and response capacity in low-income countries, and the persistent failure of the international community to invest in health systems, strengthening in the countries that were most vulnerable to epidemic emergence. The Commission on a Global Health Risk Framework for the Future, established in 2015 by the National Academies of Sciences, Engineering, and Medicine, and chaired by Peter Sands, calculated that the world had spent, in aggregate, far less on preparing for epidemic threats than the economic cost of a single major epidemic, and recommended an annual investment of approximately $5 billion in global health security as the minimum required to reduce the risk of future catastrophic outbreaks to an acceptable level. These recommendations were received seriously in the immediate aftermath of the epidemic, when the memory of the West African experience was fresh, and the political will to act was, for a brief period, relatively strong, and they resulted in the establishment of the Coalition for Epidemic Preparedness Innovations, known as CEPI, in 2017, and in increased investment in the WHO's emergency preparedness and response functions, and in progress, uneven and incomplete but real, toward the strengthening of national health systems in the countries most at risk. The scientific legacy of the West African Ebola epidemic was, alongside its human tragedy, one of the most significant scientific legacies of any epidemic in the history of medicine, generating not only the vaccine that was demonstrated to be effective in the 2015 Guinea trial, but a body of knowledge about Ebola biology, epidemiology, and clinical management that transformed the world's understanding of the virus and its relationship to human populations. The genomic sequencing work conducted during the epidemic provided an unprecedented view of viral evolution in real time, demonstrating how a single introduced strain can diversify and spread through a human population over the course of months, accumulating mutations that allow scientists to reconstruct the transmission chains of the epidemic with a precision that traditional epidemiological methods could not achieve alone. Clinical research conducted during the epidemic produced the first rigorous evidence about the supportive care interventions that most reliably improve survival rates for Ebola patients. Findings about the role of oral and intravenous rehydration, about the management of electrolyte imbalances, about the early treatment of secondary infections that had not been available from the smaller outbreaks that had preceded it. Anthropological and social science research conducted in the field provided a granular understanding of the community dynamics that shape epidemic trajectories, the role of funerary practices, the importance of trust in healthcare institutions, the mechanisms through which stigma is generated and sustained, the conditions under which community behavior change occurs most effectively. That has informed the design of subsequent epidemic responses in ways that are difficult to quantify, but are broadly recognized by those working in the field as deeply significant. The epidemic was, in this sense, a devastating natural experiment that generated knowledge at enormous cost. Knowledge that belonged, in some fundamental sense, to the communities that paid the highest price for its production, and that carries within it an obligation to be used in their service. The story of Ebola from 1976 to 2014 is, at one level, a story about a single virus moving through time. Identified in a mission hospital in northern Zaire in August and September of 1976, named after a river on a map that scientists were consulting in the exhaustion of a long field investigation, classified within a family of thread-like RNA viruses that includes only a handful of known members, studied for decades in high-containment laboratories by dedicated scientists working carefully and quietly in circumstances that most of the world would find difficult to imagine, and emerging finally in 2014 in a part of Africa where it had never before been formally recorded, to produce an epidemic of a scale and a duration that none of the prior history of the disease had suggested was possible. At another level, it is a story about the relationship between human beings and the biological world they inhabit. The ancient and ongoing intimacy between human communities and the forests and the animals that surround them, the ways in which changes in land use and human mobility and population density alter the conditions of contact between human populations, and the pathogens that circulate in the animal world, and the ways in which the social and cultural practices through which human communities mark birth and death and illness and recovery interact, sometimes tragically, with the transmission dynamics of the diseases that pass through them. And at yet another level, it is a story about institutions and systems, about the World Health Organization and its capacities and its constraints, about the governments of Guinea and Liberia and Sierra Leone, and their struggles to build functional healthcare systems in the aftermath of conflict, and in the context of persistent poverty, about the pharmaceutical industry and its incentive structures and their consequences for the development of treatments for diseases of the poor, about the international community and its capacity for solidarity and its tendency toward complacency in the intervals between crises. It is worth pausing here, in the long arc of this narrative, to observe that every Ebola outbreak from 1976 to the present, from the 318 cases of Yambuku to the 28,616 cases of the West African epidemic, has, in the end, been brought to conclusion by the same fundamental set of interventions: the identification and isolation of cases, the tracing and monitoring of contacts, the modification of burial practices, the engagement of communities in the understanding, and the management of transmission risk, and the maintenance of these measures with patience and consistency until the chain of transmission has been interrupted. These are not glamorous interventions. They do not require advanced technology or sophisticated equipment. They require people, trained, supported, trusted by their communities, and sustained in their work by institutions that understand the importance of what they are doing. Every time an Ebola outbreak has been contained in the history of the disease, it has been contained because there were people on the ground in communities doing this work. And every time an Ebola outbreak has expanded beyond what it needed to be, it has expanded because those people were too few, or too under-resourced, or too little trusted by the communities they were trying to serve, or too late to arrive, when the outbreak was still at a scale where their efforts could make a decisive difference. This is not a complicated lesson in its essential form. It is rather the kind of lesson that is simple to state and endlessly difficult to act upon consistently, because the political will and the financial resources and the institutional capacity required to support epidemic response in peacetime, in the long quiet intervals between outbreaks, are precisely the things that are hardest to maintain when the urgency of a visible crisis is not present to sustain them. The village of Meliandou, in the Gekadu prefecture of southeastern Guinea, is a small place, a few hundred people set within a landscape of forest and farmland, where the borders of three countries meet, and where the daily life of the community follows rhythms that have been shaped by the forest for generations. In the years since the epidemic, the village has been the subject of considerable attention from researchers and journalists and filmmakers who have come to document the place where the largest Ebola epidemic in history began. And the community has received some of the recognition and the resources that this attention has brought, though the depth of the loss it suffered, including the early deaths of the child, Emile, and his sister and mother and grandmother, who were among the very first to die in an epidemic that would eventually claim more than 11,000 lives, is not something that recognition or resources can adequately address or repair. The hollow tree where the children used to play, the tree where the bats roosted, and where the virus may have moved from the animal world to the human one, in the body of a small child on some unremarkable afternoon in December 2013, has been gone since 2014, burned in the ordinary business of village life, before anyone understood what it represented. There is something deeply fitting and deeply human about this fact, that the physical origin point of a global crisis was a tree where children played, that it disappeared in a fire before the world knew what it was, and that the community that surrounded it was simply living its life in the forest, in the way that communities have lived their lives in forests for thousands of years, with no knowledge of and no responsibility for the invisible complexity of the biological world that sustained and surrounded them. The fruit bat, if it is indeed the bat, goes about its life in the canopy of the equatorial forest, without knowledge of what it carries, without intention, without guilt. It is an animal of considerable grace, wide-winged and nocturnal, navigating the dark forest by smell and by the memory of fruiting trees, roosting in colonies of thousands in the high branches of the largest trees, dispersing the seeds of the fruits it eats across the forest floor, and in so doing contributing, generation after generation, to the maintenance of the forest itself. The relationship between the bat and the virus that it carries is ancient, almost certainly far older than the human presence in the forest, a relationship shaped by millions of years of coevolution, in which the virus has learned gradually to replicate within the bat, without causing the bat the kind of harm that would eliminate the host on which its own continuation depends. It is only when this ancient, quiet relationship is disrupted, when a human hand reaches into the forest and encounters the virus in the blood or the tissues of an animal that carries it, when a child plays under the tree where the bats roost, when a hunter handles the carcass of a chimpanzee that has itself been infected by the same chain of contact, that the virus finds itself in a host for which it has not been optimized, in a body that it harms severely than it harms the reservoir host, in a context where its replication and its transmission carry consequences that ripple outward through human communities, in ways that the forest and the bat and the virus itself are entirely indifferent to. The Ebola story is, in this sense, not a story of malevolence or of catastrophe as such. It is a story of contact, of the inevitable and ongoing meeting between the human world and the biological world that contains it, a meeting that has always been occurring and will always be occurring. And that carries within it the possibility of both wonder and sorrow in measures that human beings have not yet learned, and may never fully learn, to predict or to control. Over the decades that followed the initial identification of the Ebola virus in 1976, the world's understanding of the disease, its biology, its epidemiology, its social dimensions, its place within the broader landscape of global health, grew in ways that the scientists who named the virus after a river on a tired afternoon would not fully have predicted. The research programs that produced this understanding were sustained by the dedication of scientists in many countries, by the courage of healthcare workers in the affected regions, by the participation of communities in field studies and clinical trials, and by a degree of international collaboration across political and institutional boundaries that is, whatever its limitations and its failures, one of the more genuinely hopeful aspects of the human response to epidemic disease. Peter Piot, the young Belgian scientist who first looked at the Ebola virus under an electron microscope in Antwerp in October 1976, went on to a distinguished career in global health, leading the Joint United Nations program on HIV/AIDS, and later directing the London School of Hygiene and Tropical Medicine, and remaining throughout his life associated with the history and the ongoing challenge of Ebola and its related diseases. Carl Johnson, who named the virus, continued his career in tropical virology, studying hemorrhagic fevers across 4 continents, and contributing to the institutional knowledge of the field over decades of careful, patient work. The scientists who came after them, the virologists and epidemiologists and clinicians and anthropologists and public health specialists who have engaged with Ebola through the 40 years and more of its known history, have built, collectively, a body of understanding that is not yet complete, but that is immeasurably richer than what existed in the autumn of 1976, when a blue thermos arrived at an institute in Antwerp, and a young scientist looked through a microscope and saw, for the first time, the distinctive and unfamiliar shape of something that had no name yet in the human lexicon of disease. The West African Ebola epidemic ended not with a declaration of triumph, but with a quiet, gradual, uneven subsidence. The case counts falling, the treatment centers closing, the international staff departing, the communities beginning, slowly and incompletely, the long work of grief and reconstruction and re-engagement with the ordinary challenges of daily life that the epidemic had interrupted. The survivors of the epidemic, those who had been ill and recovered, those who had lost family members and neighbors, those who had worked in the responses and carried the experience of that work with them, returned to their lives carrying the weight of what had happened in ways that would continue to shape them for years and decades. The healthcare systems of the 3 most affected countries, fragile before the epidemic, were in many respects more fragile after it, and the work of rebuilding them, with the residual international support that remained after the emergency phase of the response concluded, and the world's attention moved elsewhere, was slow and difficult and insufficiently resourced. The children who had been orphaned by the epidemic grew older in communities still marked by loss. The survivors who had been stigmatized continued, in many cases, to navigate the social consequences of their survival long after the formal end of the public health emergency. And the forests of the Mano River region continued to exist, ancient and complex, and largely indifferent to the human events that had occurred at their edges, sheltering within their canopy, the bats and the primates, and the 10,000 other species that constitute the living community of the equatorial forest, and within that community, almost certainly, the viral passenger that had moved from the forest into the human world in December 2013 and had not, in all probability, departed. And so the history of Ebola, from the quiet forest of northern Zaire in the summer of 1976 to the forest borderlands of southeastern Guinea in the winter of 2013, and the long, sorrowful arc of the epidemic that followed comes to rest here, not in the manner of stories that resolve cleanly, with a conclusion that ties all threads together and leaves nothing unfinished, but in the manner of histories that end, because the telling of them must end, while the events they describe continue to unfold in time, shifting slowly and patiently beyond the last paragraph, beyond the last sentence, beyond the last word. The virus persists in the forest, in all probability, as it has persisted for a time far longer than any human memory can reach, waiting in the quiet reservoir of whatever animal species has agreed, through the long negotiation and evolution, to carry it without being destroyed by it. The communities of Guinea and Liberia and Sierra Leone and the Democratic Republic of Congo carry their histories of illness and loss and survival and resilience in the ways that communities always carry their histories, woven into the stories they tell and the practices they maintain and the knowledge they pass from the older to the younger. The scientists continue their patient work in the laboratories and in the field, building the understanding that the world will need the next time the virus finds its way from the forest into a human community. And here you are, in the quiet of this moment, breathing slowly and easily, your body soft against whatever holds you, the long history of a distant illness drifting through your awareness like a leaf, drifting downstream on the slow current of a dark river, moving gently past and away. The Ebola River is very far from here. The forest of the Congo Basin is very far from here. The past is very far from here, held safely in the record of what has been, in the careful accounts of the scientists and the physicians and the community workers and the survivors who lived through it and bore witness to it and gave us, through their effort and their sacrifice, the understanding that we carry forward. Let the names and the dates and the places settle quietly into the soft background of the night. Let the story rest. Let the river move on without you, as rivers always do, carrying everything away downstream, toward the sea, toward the dark and the quiet and the dreamless, where all history eventually arrives. You are here. You are safe. You are breathing, and the night is very gentle and very still and very kind, and there is nothing left to do but let it hold you.